We have reviewed the microscopic anatomy of the heart. Let's look at how those cells function. You have to remember that muscle cells contract in response to an electrical excitation. The excitation itself is a change in polarity along the cell membrane, whereas a contraction of the muscle cell occurs due to cross-bridge formation or interaction between thick and thin filaments. And of course, we have studied that all in great big details last term, so this is just a quick review for you, right? So how does it work with the heart muscle? There are two different types of heart muscle cells, or cardiomyocytes. There are cells that contract predominantly, and they're called contractile cells. They have other names as well. And then there are cells called pacemakers. They're also myocytes, but their job is predominantly to generate an excitation and spread it around the heart. So let's first look at the contractile cells. The main job for these cells is to contract. Under very special circumstances, they are capable of generating their own excitation, but it's very, very slow rate, and in reality, they practically never get to do that. So, in essence, they contract when they're stimulated, and that stimulation from outside results in a change in polarity or action potential on contractile cells. So let's look at the phases of action potential. On this diagram, you see the graph that represents the action potential, and it represents changes of voltage in time. So the following phases will be discussed: a resting state, a rapid depolarization, partial repolarization, plateau, repolarization, and resting potential. So let's look at it in a little bit more details. To start with, I want to remind you that concentration of ions inside of the cell and outside of the cell is not the same. Sodium is predominantly found outside of the cell because there's more sodium on the outside than on the inside. If the membrane of the cell is permeable, sodium would want to go inside the cell. Potassium is predominantly found inside the cell. And so if the membrane is permeable for this ion, the potassium will tend to leak out of the cell. And the last player that we want to discuss is calcium. Calcium is also found in large quantities outside of the cell. If membrane is permeable for calcium, calcium will tend to drift into the cell. Now you also have to remember that because all of those ions have a charge, membrane that has a hydrophobic layer would not be permeable to those ions in their normal state. So how do those ions get in and out? Well, through the channels. The channels that will affect the polarity of membrane are regulated by voltage across the membrane or potential difference across the membrane, and therefore they're called voltage-gated channels. So obviously we will be dealing with sodium voltage-gated channels, potassium voltage-gated channels, and calcium voltage-gated channels. You also have to remember that at rest, the cell membrane is slightly permeable to potassium, and potassium leaks out of the cell, taking its positive charge with it, which leaves the inside of the cell relatively negative. So at rest, membrane has a voltage across it of about negative 90 millivolts. So it has a relatively negative charge on the inside and relatively positive charge on the outside. Make sense? So in the very first phase of action potential, membrane becomes permeable to sodium. Sodium voltage-gated channels open, and of course, because there is a concentration gradient, sodium rushes into the cell. But let's look at this very carefully. Is that the only reason why sodium wants to come in? Look, membrane has a relatively negative charge on the inside, and sodium has a positive charge. So that also adds an electrostatic force that drives the sodium into the cell. As sodium enters, it brings its positivity with it, and membrane becomes less and less negative on the inside and less positive on the outside, and eventually reaches the point where it changes its polarity, hence the name depolarization. Let's look what's going on the graph. Change in polarity, right? You can see that now the membranes charge positively on the inside and negatively on the outside. That's because sodium has entered the cell, correct? When sodium channels close, potassium channels open, and that signifies the partial repolarization phase, of phase 1. In this phase, potassium, that has a permeability or green light to cross a membrane, leaves the cell. It leaves the cell again for two reasons. One, because I have a concentration gradient. I have too much in the cell, very little outside. And second, electrostatic force again drives this positive charge to the negative charges on the outside of the cell. What do you think will happen to the polarity of the membrane? It will become less and less different from inside to the outside, right? And we will see repolarization or restoration of the potential. Make sense? Now, when membrane gets close to zero, not only permeability of potassium exists, but also membrane becomes permeable to calcium. And of course, since we have concentration of gradient for calcium, calcium enters the cell. Calcium only enters down the concentration gradient. Notice that there is no electrostatic force for calcium. So what happens overall to the polarity of the membrane? I have positive charge leaving and positive charge coming. The polarity of a membrane does not change. So we will have a flat line here, and that's what we call plateau. Calcium channels only open for a fixed period of time, and when they close, permeability of the potassium is the only one that remains. And of course, since there is still a concentration gradient for potassium, it continues leaving the cell, bringing the membrane potential back to the resting state. So that phase is called repolarization. Here you can see that membrane has restored its polarity. It is positive on the outside, negative on the inside. Make sense? So we are back to where we started with the voltage across the membrane. However, there are all these ions that have been escaping or entering the cell. They have to be placed back to where they belong so that we can start the new action potential. That occurs during the resting potential, where pumps return ions back to their original place, but the membrane potential remains the same. And upon the completion of this phase, we are ready to start a new action potential. Let's recap what we have just said. At rest, membrane is at about negative 90 millivolts. During the initial phase, which is, by the way, phase O, I don't know who decided to count that way, 0, 1, 2, 3, 4, but that's how we traditionally count the phases. The first phase is rapid depolarization, and it is due to the sodium influx. Next phase is early repolarization, and it is due to the potassium outflow. Next phase is a plateau due to the potassium outflow and calcium inflow. Repolarization is due to the potassium outflow only. And the resting potential is observed with no changes in polarity, but with the restoration of ion concentration inside the cell. Now that all talks about the membrane events, changes in polarity across the membrane. Remember that the contraction is actually intracellular event. So we now have to relay this excitation of membrane to the interaction between thick and thin filaments. And of course that is done via a special messenger, calcium. Just like in skeletal muscles, we have transverse tubules that extend down into the cell and excite sarcoplasmic reticulum. But also the calcium that enters the cell during the plateau is in part the calcium that will activate troponin and therefore make thin filaments available for the interactions with the thick filaments so that the cross bridges can be formed. So again, we are heavily relying on the calcium that enters the cell during plateau for the cell to start contracting.
Cardiac contraction is initiated when extracellular calcium moves into the cell across the voltage-dependent calcium channel. The calcium that enters the cell triggers calcium release through a process known as calcium-triggered calcium release. Calcium that is released from the sarcoplasmic reticulum binds to the actin-thin filaments, thereby initiating contraction of the sarcomeres. At the end of the contraction sequence, calcium comes off of the myofilaments and is actively transported back into the sarcoplasmic reticulum by the calcium pump in its membrane. Calcium is also transported out of the cell into the extracellular space by a membrane-bound pump sodium-calcium exchanger, which is not shown here. Cardiac contraction is brought about by interactions between the actin-thin filament, shown in yellow and purple, and the myosin cross bridges that project from the thick filament, shown in red. In relaxed cardiac muscle, the conformation of troponin and tropomyosin prevents actin from interacting with the myosin cross bridges. As a result, actin is unable to interact with the myosin cross bridges containing bound ATP. In activated cardiac muscle, calcium binds to troponin C, which then shifts troponin complexes and tropomyosin to an active conformation that enables actin to interact with the myosin cross bridges. Myosin ATPase on the myosin heads then hydrolyzes ATP, thus providing the requisite energy for the myosin cross bridge to interact with the thin filaments and pull the thin filaments towards the center of the sarcomere in a ratchet-like fashion. In this sequence of the normal heart, we have reviewed the process of excitation-contraction coupling.
Let's see if we can do a few practice examples. Depolarization in contractile cells is due to the sodium influx. Very good. Cardiac contraction. When do you think the cell will start contracting? Depolarization, early repolarization, plateau, final repolarization, or resting state? The answer is plateau, and that's of course because calcium enters the cell only during plateau.